everybody. Welcome to the live stream tonight. We're going to go ahead and just dive right on in. I've got my worksheet ready to go, and hopefully you guys do too. Let me just go ahead and switch the screen over to the worksheet where we're going to be going over chromosomal basis of inheritance today. So more deep diving into genetics. So we'll just go ahead and start with that first problem here. If at any point you have any questions about any of the stuff that I'm going over today, just feel free to write it down in the chat here. So first question is asking about an organism with the genotype AA, BB, CC. First initials or first letters are big letters. The second ones are all small letters, crossed with an organism that has the genotype 2 big A's, 1 big B, 1 small b, 1 big C, 1 small c. Assuming all genes are on separate chromosomal sets, meaning that they're not linked. And it's asking here the probability, what is it, that any of the offspring will have the genotype all big A, all big B, all big C. And then it gives a hint to consider 6 possible types of autosomal crosses, determine the individual probabilities of getting 2 big A offspring from the monohybrid cross, then do the same to determine the probabilities of getting 2 big B offspring and 2 big C offspring. Then multiply the probabilities together. So it's a lot of words for really simple stuff that they're looking for, which is here is A, and I'm going to do them in different colors. This is AA, and this is BB, and this is CC. And they just want the fractions for each one. AA is going to be 1/2, BB is going to be 1/4, CC is going to be 1/4. Therefore, the one that is the combo of AA, BB, and CC, and if this little like box thingy, it just came with my download of the worksheet. That is then going to equal the following, which is 1/2 times 1/4 times 1/4, which is going to equal 132. And then we'll move on. Probability that any of the offspring will have this different genotype, which is big A, big B, little b, and then 2 little c's. So from here, you're going to do A and then small a, big B, small b, and then 2 small c's. And these are going to equal 2 different fractions, 1/2, 1/2, and 1/4. So then that means for the AA, BB, and the 2 little c's, that is the equivalent of 1/2 times 1/2 times 1/4, and that is going to come out to 1/16. And then consider this large cross. I'm not going to say all of it. And it's asking again for a little bit more math. And what it's asking for is the following: 1/2 times 1/2 times 1/2 times 1 times 1/4 is going to equal 132. And what I'm going to do here is I'm going to color code the probability here. So this is equal to this, 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 and this is equal to this. And then moving on, what's the probability here? I'm just going to move this a little bit down. There we go. What's the probability any offspring will have this even bigger genotype where it's all these dominant, aka big letters, and then 2 small e's, 2 small letters at the end? And that'll be the following: 1/2 times 1/2 times 0 times 0 times 1/4. And since there's a 0 in there, it's going to equal 0. Then the next question says, in fruit flies, most common eye color is red. Mutation or allele of the gene for eye color produces white eyes. The gene is located on the X chromosome. What is the probability here that a heterozygous red-eyed female fruit fly mated with a white-eyed male will produce any white-eyed offspring? So this is going to be a big table. So let me write the what it's looking for on the side. And again, I'm going to color code it. So we have X, W, plus X, W. And that is going to be, all of this is going to be times, and I will make it smaller so this can be actually read. Let's see here. I feel like there's something missing. Yeah, I'm going to erase this. I don't really like the way this looks. I'm going to actually just draw the table. This is just going to be easier to understand if I do it this way. And then here we go. And then here are all the crosses that will come with it. And that what you'll say here is that half white-eyed offspring, and then half I'm going to make that smaller because there's a particular word I'm looking for that I forgot to mention here. So the first half is going to be male, and the other half is female. And then it says here that the probability that the mating in part A will produce any white-eyed males. This is probably going to be 1/4, okay? And then it's asking here, probability of producing any white-eyed males is 1/4. I think that might be a typo because I think that you were supposed to come up with that answer yourself, but it's okay. The next one is asking about a heterozygous brown-eyed human female who is a carrier of color blindness marries a blue-eyed male who is not colorblind. It is a sex-linked trait, and you're assuming the eye color is an autosomal trait and that brown is dominant over blue. What is the probability that any of the offspring produced have the following traits? So I'm going to go ahead and write down the fractions for each one, aka the probabilities. So 1/2, 1/2, 1/4, 1/4, 1/2 times 1/4 equals 1/8, and 1/2 times 0 equals 0. And that's because you're using the following crosses, which is BB, that is X plus XCB, female, times small b, X plus Y, male. And the crosses that you're going to do are the following, and I'm going to color code them. BB, oops, times, and then X plus B times X plus and Y. Okay. Probability that any of the males are going to be colorblind is going to be 1/2. And just wanted to note that this question is just asking only about the males, not all of the offspring. And if we were to look at all of the offspring, we would find 1/4 will be colorblind males. So we can go ahead and write that down. Oops, looking at all offspring, 1/4 will be color blind males. Okay. And then it's asking, why do males show sex-linked traits more often than females? And it's because, I'm going to zoom in a little bit so I can write just a little bit easier. And it's because males have one X chromosome. Which carries more genes than the Y chromosome. Females have 2 alleles. Have 2 alleles for every gene. On the X chromosome. And that's what my little squiggly thing is, that it's an and. And they have not a, the recessive phenotype when both X's carry the recessive allele. And then in contrast, sorry, this is a lot of text. Males need to have only one recessive allele to show the recessive phenotype. And hopefully my writing makes sense. So now we'll go ahead and zoom out and then scroll on down to the next thing. This one is now about solving problems when the genetics are unknown. And it's saying that Mendelian genetics allows us to determine the theoretical probabilities associated with normal transmission of autosomal and sex-linked genes during reproduction. And we'll skip down the rest of the text to go down to what's asking for with the 2 problems presented below, which is in A, it's a wild population, aka the phenotypic characteristics of a wild population of fruit flies that were trapped randomly on a remote island Meanwhile, with B, it's cross 1 and 2, etc. The phenotypic characteristics of offspring from a controlled cross. The phenotypes of the parents are indicated after each cross, e.g., cross 1, male Ambler times female wild type. So, scrolling on down, this has already been pre-populated for you. And it's saying to analyze the results in each cross and answer the questions that follow. So, it says here, what does cross 1 tell you about dominance versus recessiveness of the alleles? Again, I'm going to zoom in just a little bit so that I can write a little bit better. And it's saying here, it is impossible to determine if one is dominant over the other because you get equal numbers of both phenotypes. All right, next one. What does cross 1 tell you about the placement of the alleles on autosomes versus sex chromes? And it's saying here, again, impossible to determine if the, I tend to jump into cursive to save time. If the alleles, or the allele, is on an autosome or sex chromosome, due to the number of males and females in each phenotype being the same. All right, moving on. Moving to now cross 2, which is saying it's a female Ambler and a male wild type. It's asking here, what does cross 2 tell you about dominance versus recessiveness of the allele? Indicates, and I just want to zoom in because it's just easier for me, indicates that, indicates that the Ambler is recessive because all males are Ambler and all females are wild type. And then what does cross 2 tell you about placement of the alleles on autosomes versus sex chromosomes? And what we'll write down here is, let's see, the alleles, are on the X chromosome, and then here's the parent cross. And then X, AM, X, AM, female, and X plus Y. All right, moving on to problem 2. 2 different tables here that are talking about these different types of phenotypes: bonical, bifocal, trifocal, spinner, shiny, etc., etc. And then it's asking again, what does cross 1 tell you about dominance versus recessiveness of the alleles? And again, zooming in, just to give space to write, indicates MT and BT, are codominant. Indicates 1, 2. TR is heterozygote, or is the, move that to the side, is the heterozygote. And then, let's see, let me remove that word, not indicates, doesn't indicate. If SP or SH are dominant. Because, I'm just going to move this to the side. Sorry for the errors, guys. Because they appear in equal numbers. Now, what does cross 1 tell you about the placement of the alleles on autosomes versus sex chromosomes? That MT and BT alleles are autosomal because all males and females are TR codominants. So this is number 1. And then number 2 is that it's unclear if SP and SH alleles are autosomal versus sex linked, because the numbers, well, mainly the male-female numbers, with each trait are the same. Okay. Moving on to another one. Woof. Lot of stuff going on in here. But I think we're doing good on time. Yeah, we're doing pretty good on time. We're already halfway through. So, and I think we just have this page and then this. Okay, so 2 more questions about this table with these alleles, or these phenotypes. So what is that phenotype? They are alleles. Sorry, I've been saying the wrong word. Genetics is very hard for me. This is not my favorite topic in biology. I'm pretty sure I've said this before. What does cross 2 tell you about dominance versus recessiveness of the alleles? This confirms, MT and BT alleles are codominant. And then you're going to say here the following: MT and MT times MT-BT is going to turn into MT, MT, and might as well use that line for my benefit, MT-BT offspring. Then it says here that SP and SH are recessive. Well, sorry. That was the wrong wording. Sorry about that. It's just that SH is recessive. Nothing else. Because that goes down here for what does cross 2 tell you about the placement of these alleles on autosomes versus 6 chromosomes. So we're going to make another table again. Try to make it as accurate as possible. I'm not going to color code again. X, SP, and then down here is Y. And down here is X, SP. XSH, and again, I've made the table too big. Again, sorry about that. Then we'll say XSP, 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 Y, XSP, XSH, and X, SH, and Y. And what I'm going to do is that for each one of these, I'm going to assign them the following. And that this down here is this. So then this is SP males, SP females, SH males. And that all of this is basically to represent that we should see we put a star right next. We should see 0 SH females. And then the other thing we want to say in a star is ratio of the others are 2 SP females equals, 2 SP females to 1 SP males to 1 SH males. Okay? Now this is an analysis of the pedigree, and if you are planning on taking more genetics, you really got to be familiar with this. You will see it again and again in genetics. This diagram below shows a pedigree of 3 generations in a family. The black circles and squares indicate persons with a genetic disorder. Square indicates a male, and the circle indicates a female. The 2 males in generation 1 are siblings. So now from here it is asking, looking only at the generation 2 offspring of the 2 generation 1 brothers, what can you say about the genes controlling the genetic disorder? Is the disorder caused by a gene that is dominant or recessive, autosomal, or sex-linked? So first thing you're going to say is that gene, and again, I'm just going to zoom in. It's just easier for me if I do that. Gene is most likely dominant, which means gene is possibly autosomal or sex-linked. It could also be recessive. But for that to be true, the two brothers would both have to mate with a heterozygous female to produce the Gen 2 offspring. What additional information do you gain from examining the Generation 3 offspring? And from here, you're going to say, in Gen 2, two affected individuals produce one unaffected male offspring. And just a little bit information. If the disorder was caused by an autosomal recessive gene, all the offspring in this cross would be homozygous recessive, and have the disorder. And because one male does not have it, the disorder is probably, well, it must be caused by a dominant allele. Okay, moving on. How can the mode of inheritance be determined exponentially? Outline the experimental crosses that you would need to make to solve each problem. 3 new traits have been discovered in a population of fruit flies: tapping, single stripe, and angular. The positions of the 3 genes in the chromosomes are unknown. Given 2 pure breeding, aka homozygous lines, and using an initial cross of normal females with tapping, single stripe, angular males, describe the appropriate genetic experiments needed to establish whether any of the traits are caused by genes that are autosomal or sex-linked, for example. So let's see here. Let's go ahead and make, actually, is this the last page? It is. Wow, we are going to be finished early tonight. And I hope y'all are okay with me finishing early, so that way you can just get this done and submit it and be over with it. So let me go to the next page and I will start drawing out the table that will be looking good on this piece of paper. Okay, here. So let's see here. Let's just start filling in stuff up top. Okay. And I'm going to start making little rows up top here. Okay, so in this section we're going to have A, and then over here we're going to have A and A, and then big, I'm actually going to make that a bigger line, big thick line, probably a little too thick, but okay. Just to kind of make it obvious that they're not the same, that's different. Okay. And then the other thing that I'll do is then do the next one, which is going to be A. Oops. Now it's just too big. A, and then a line, and then big A, and a small a. And again, I made it way too big. Story of my life. Okay. Now we'll just start putting things in here. So we're going to have A, A, big A, small a, and then big, oops, A, and then I'm just going to do another table down here. Sorry about that. One. I feel like this is just going to take the most time, honestly. And then let's go ahead and start making the other table. Let's just make that big. Again, big thing here, just kind of making sure that you know this is not the same, but that they're similar. And then we'll do the same over here. And now we'll do the other one. So let's do this one in purple. So we'll make this one here XA. And then I'm just going to start pre-filling in the XAs. Oh, not that A. Sorry. Good job, me. And I'm going to choose a different color up here. Let's go with, let's do that one, I guess. Or, let's go with a nice dark red. Sure. And this one will be XA and Y. Y, X, big case A, X, big case A. And then let's do a color over here for the last one. So this one will be XA, and this one will be Y. So let us do the XA, XA, and Y, and Y. And the last ones are going to be this color here. And this is X big case A, X big case A, X big case A. And then XA, XA, XA. Okay. And then what you'll write on the side here is that the crosses were the following. A, B, C, C times A, B, C, C. And I'm just going to make that smaller. There. And I'm just going to zoom in a little bit just to make it look a little better. And that all F1 times A, B, C, C. And make these smaller as well. And then you will say here F1 crosses with homozygous recessive mates, and they will be underneath here as the A, B, C, C times A, B, C, C. And again, let's just make that a little bit smaller. There we go. Excellent. So that's what your table looks like for here. And then we just want to explain a little bit as to what all of this means. Because again, it's like, wow, I just spent a lot of time drawing that table. What does this mean? It's asking the positions of the 3 genes on the chromosomes are unknown, given 2 pure breeding lines, and using an initial cross of normal females with tapping single-strap angular males, describe the appropriate genetic experiments needed to establish whether any of these traits are caused by genes that are autosomal or sex-linked. So here's the table that you made, and that what you're asking here is that the first, like, just a little bit extra steps. So the first step is to mate normal, homozygous, females with tapping, single stripe, angular males. And then you can go ahead and add a little thing down here that says F1 phenotypes. So the F1 individual phenotypes will indicate which alleles are dominant. And then I'll fix that so it looks a little bit better. And then what you're going to say here is then mate F1 males with homozygous females. Homozygous recessive, sorry. I forgot that word, homozygous recessive. Homozygous recessive females for all 3 traits. And then you're going to do the following: mate F1 females with homozygous recessive males for all 3 traits. So there's that, and then I'm just going to get all of this and make it just a little bit prettier. There we go. That just looks a little bit better, I think. I'll even move that over here. And then this up here. With this over here. There we go. The last thing you'll do is examine phenotype ratio for each trait. And compare with the whole ratio. For each sex. Okay. And hopefully that looked like it made sense. Again, I'm just going to make that smaller. There we go. Again, here's what it looks like. And again, it doesn't have to be a big block of what I made. What I wrote down also counts, but figured having a little bit of a table helps you understand it a little bit better. All right. Then the last one is asking linked on the same chromosome or unlinked. And again, it's asking about the normal with this tapping single-strap angular males. Appropriate genetic experiments needed to establish whether any of these traits are caused by genes that were at first autosomal or sex-linked. Now we're down to sex-linked on the same chromosome or unlinked. So let's go ahead and write out the steps first. Number 1, we will say if not linked genes, expect probability with given set of phenotypes. Like normal, stripe, angular. As a matter of fact, I'll even add, this is an example. And that you're expecting these given sets of phenotypes to be equal to product of the individual probabilities for each occurring separate crosses. And then you're going to say here, if genes not linked, expected to see one-half normal, one-half one-stripe, and then one-half angular. And then you'll say here for 3, probability of all these, all these characteristics showing going to be the following. It's going to be 1/2 times 1/2 times 1/2, which equals 1/8. And then you'll say for if linked, if genes linked, i.e. A and B, BF1 cross becomes the following. I'm going to rewrite that word cross. Becomes the following. It becomes AB times AB, which turns into 1/2 AB, B, and 1/2 AB. And then the other one is going to be your CC times CC, and that turns into your 1/2 CC, and then the 1/2 CC. So I'm going to move that guy over here. So lots of words, but we have room to put in a tiny table. I say tiny. This is what the table's going to look like. I'll make a thing up top here. I'll put a little section right here. And another section right here. This is going to be your 1/2 AB slash AB. 1/2 AB, AB. So again, this is just a table version of what we just all wrote out, or what I just wrote out. And then 1/2 CC and 1/2 CC. And now for all the combos. AA, BB, CC, 1/4 AA, BB, CC. Over here, 1/4 AA, BB, CC. And then 1/4 AA, BB, CC. And that should be it. So I'm going to move this a little bit over to the side over here. And move this over here. And then I'm going to move this all the way up here as part of the table. And that was it, everyone. Okay, I said we would end early, and it's a little bit early, but not too much. So we have a couple of minutes left. I'm going to go ahead and stop recording, so if you're watching this on YouTube, thanks for watching.